My name is Oliver, and I am the Genomics and Single Cell Manager at Paediatric, Bioskryb's French distributor. So in a few moments, I will hand over to Luca Mazzitelli, EMEA Manager at Bioskryb, who will give a short introduction to Bioskryb's single-cell genomics and their proprietary PTA technology, so for primary template-directed amplification. And then Alexander Stimmers from the Princess Maxima Center for Pediatric Oncology will present his results obtained with Bioscribe technology for his single-cell whole genome sequencing studies in the context of pediatric Burkitt lymphoma. So, Luca, I'll just let you
Thanks,
start your
Olivier.
presentation.
Thanks as well for your partnership and your help in supporting and making this event today. So let me share my presentation. I will have a brief technology overview about Bioskryb. Let me put the pointer here. That's it. So my name is Luca Mazzitelli, and I'm the channel manager EMEA at Bioskryb Genomics. I support distributors all over Europe. And today I'm going to present how leveraging single-cell DNA inform the multi-omics in discovery research. So the mission of Bioskryb Genomics is really to help researchers to understand this immense cellular heterogeneity that drives complex biology and also tissue biology and disease. So in these figures, what we see is the mosaicism that actually increase as we age. And this happens, so this accumulation of mutation happens not only in healthy tissue and not only in diseased tissue, but also in healthy tissue. We know that RNA and all epigenetic changes are quite a dynamic environment, but it's also important to acknowledge that our genome is not static. Therefore, recognizing and decoding this genomic diversity is essential to develop the next generation therapies and diagnostics. And we do this at single cell level because each cell has its own potential. What you can do when you perform a single cell analysis is several kinds of application. Like in PGT world, in pre-implantation genetic testing, where you can predict the viability and the health of potential offsprings. Or you know that one immune cells can be recruiting other inflammatory cells into tissues. Or when you are in a situation of cancer developing into a more invasive cancer, this comes from one clone that actually is probably escaping the treatment and can drive tumor growth. Or single cells can have a specific mutation profile, can carry mutation that can be used to trace back then the origin of that cells. So single cell sequencing is really about clarity. What we see here on the left is bulk sequencing that gives us an average data, so a kind of noisy data where, for example, real through somatic mutation, these are in greens, are going to be confused with sequencing or amplification factors. When we go and we move into the single cell sequencing, this changes the game a lot. We really obtain a digital sequencing. So a true heterozygous mutation in this figure shows in the 50% of the reads from an individual cells. So this is the kind of level of precision and confidence that helps us to survey the genome even for rare events. How we address this? So where is the innovation in Bioskryb? The innovation is in our whole genome sequencing, in whole genome amplification, which we call the primary template directed amplification. The way that we do this at Bioskryb is through PTA. So PTA works as we leverage an isothermal polymerase that binds short random primers that are hopping at multiple locations of the primary template, so on our genome. It starts an amplification, the polymerase, until it encounters a terminator base, which terminates the extension. And the results, it's down here, is a collection of amplified fragments in the range of 250, 1,500 base pair. What is important to notice here that our chemistry favors the primary template. So the Phe29 polymerase goes back to the primary template and starts other cycles of amplification, generating other amplicons. And this is really important because in this way, what we obtain is a real uniform and comprehensive amplification of the whole genome, in contrast with multiple displacement amplification, or MDA, which is an exponential amplification where a lot of areas are actually not going to be covered. One other advantage is that because we are not reamplifying those amplicons, the likelihood of reamplifying potential errors, it's minimal because our is a quasi-linear amplification. So this allows us also to decrease the amount of sequencing that we will use later in the whole genome sequencing step. We have two solutions, two products that are going under the name of Resolve DNA for whole genome amplification, and Resolvome, which is our multi-omic solution to perform whole genome sequencing and RNA analysis of the transcriptome from the same single cell. And they're both based on PTA. The workflow is from cells, from isolated cells to libraries. It can be done in one day. There are several QC steps and stop points. You can even, stop the reaction one day and restart it the day after. We are NGS platform agnostic, and we also provide Base Jumper, which is our software or that works both in cloud or as local. So we provide the pipelines that can be downloaded locally to perform the data analysis on the copy number variation, on the single nucleotide variants, and transcriptomic analysis. We also developed demonstrated protocols. If you are, not into analyzing the whole transcriptome, the whole genome, we also are compatible with enrichment for exome and panels. How the technology looks like, this is the workflow for Resolve DNA. So workflow starts with after cell collection. So you will need to work with fax to sort the cells and really to plate each cells in a single well. After cell selection, you can perform cell lysis and whole genome amplification. And then we provide also the reagents for library preparation. The workflow, as I said, it can be done in one day. The important thing is that PTA is pretty flexible in terms of input. So you can work either with single cell, for example, in neurology, where you can work with a single nuclei, or even with what we call pseudo bulk, like a very low number of cells. If you are not able to really select and sort single cells, this is still a technology that can be used with a small amount of cells, or even with low template, ultra low template DNA. And after whole genome amplification, actually you can perform library preparation for sequencing, but it can be the output can be also used for microarray or even for PCR input. Let's see kind of how resolved DNA, for example, is used, has been used to drive discovery. We have several fields of application from hemato-oncology to neuroscience. So in this first case, in this model of acute myeloid leukemia, we were able to identify single nucleotide variants difference between parental and resistant cells line, including a point mutation of the FLT3, which are known to drive resistance. Or in primary breast cancer sample. Here we were able to identify losses in chromosomal regions associated with common tumor suppressor. And what you can notice here, those are the profiles of actually single cell. And here you see the profile of a diploid cell, while here you can see the profiles or other cells which are not epithelial cells, but they have like a combination of copy number variation in various tumor suppressor. In neurology, we also have several applications over here. So quite a lot of researchers that are using resolved DNA in this area to address neurodegenerative disease. So usually, as we say, as we age, we accumulate mutations. So to identify the somatic mosaicism, this is a perfect solution to identify the somatic mutation in individual neurons. Other type of application can be in gene editing approach. So during cell and gene therapy, when you are actually using a CRISPR-Cas9 approach to perform a gene editing, and with this solution, you are able to precisely identify not only the on-target events, but also all the off-target events that maybe they are not going to be identified and characterized with bioinformatic analysis. And as I mentioned before, is also an application for pre-implantation genetic testing, where you work with precious sample biopsies. I want to give you just a snapshot of this preprint that was released recently in acute lymphoblastic leukemia. This is from the Dan Landau New York Genome Center and Chuck Goad from Stanford. In this paper, we know that over 80% of the children with acute lymphoblastic leukemia can be cured by treating them with multiple chemotherapeutic agents. But the question is, what are the rare clones that are escaping the treatment? So they performed three approaches to uncover the genomic heterogeneity: bulk error corrected sequencing, single cell exome sequencing, and resolved DNA for single cell whole genome sequencing. And they find out interesting results, like they were able to uncover rare clones previously undetected, linked to treatment resistance, and they created phylogenetic analysis to track actually the clonal evolution during therapy. So the key takeaway, there are so many other details in this preprint, but the main takeaway is that PTA reveals hidden layers of mutational complexity and clonal evolution, providing a sharper tool to understand resistance and relapse. Moving on to our multi-omic solution, so we have Resolvome, where you can obtain DNA and RNA analysis from the same cell, and this brings basically higher resolution. You know that with a single cell RNA-seq approach, you are usually able to profile your cells based on the transcriptomic profile and characterize and call those cells with a name. But what if you can add on as well the copy number variation change or the mutational signatures and have a better picture of what's happening actually in your samples? So you can determine whatever genomic alteration results in change in gene expression and in transcript structures, or you can move from gene expression and find out what are related to some genes that they have altered expression profile, what are their what is the influence from the genome. You can do this using Resolvome. Here is how the workflow looks like. Again, first step is a cell collection. And then the workflow looks a little bit more complex than the resolved DNA, but can still be done in 1 day or a couple of days. First, you do a gentle alkaline lysis with reverse transcription, and then after nuclear lysis and whole genome amplification via PTA. Then you can split the two fractions, so the amplified, the cDNA from the DNA, and start library preparation until data analysis using our software-based jumper. I just want to give you an idea on how Resolvome, for example, was used in this paper. So the anatomical origins of some human brain cell subtypes remain unknown, and the challenging to follow human brain development by conventional approach, like repeated sample, it's unethical. So the key question here was, can the analysis of genetic variation in individual cells within specific cell types reveal anatomical origins? So in addition to other cutting-edge approaches, the research teams utilize Resolvome on 191 sorted nuclei that allows them to perform cell type characterization based on the transcriptomic profile and address the mosaic variants genotyping in the same cells, allowing them to uncover the lineage relationship governing the development of human forebrain. I just want to highlight that our solutions are also automation compatible. So we have Resolve DNA and Resolvome that can be automated for the 384 reactions with Tican Uno. We provide the scripts for this. We also provide the script for Resolvome 384 that can be recently automated on the Integra Assist Plus. So this allows you, of course, to have better reproducibility and to take more time to think about your research and get the help of the automation to perform the Bioskryb workflow. One last slide that I want to highlight is that in case you have some specific needs and you want to have some kind of custom projects, well, we have specialized teams based in US. In our headquarters in Durham is the Resolve service team, where we provide services that can include analysis of cells, nuclei, or low input DNA. They can perform Resolvome or Resolve DNA workflow. We also have recently launched the beta program for DNA long reads. And also important, we do provide specific bioinformatic support and services for the data analysis. So please get in touch, and thank you for your attention right now. These are our contacts, and if you have any questions, we have the Q&A chat where you can place your question. And stop sharing and leave the stage to Alex.
Thank you to both Bioskryb and Paediatric for inviting me today to present some of our work. I'm just going to quickly put out the presentation. Could you just confirm that you can see my slides? Yeah?
Yes.
Yeah, perfect. Yeah, so a couple of weeks ago, I've been asked to present today, and initially I thought it was going to be a predominantly French audience. And so I was looking back into my high school notebook to refresh my French memory. But luckily today it's quite an international group here, so luckily for me, I'll be presenting in English. So today I want to just talk to you about how we're using PTA to solve some of our questions. And so my talk is titled The Clonal Evolution of Pediatric Burkitt Lymphoma Through Time and Space. Just to give you an introduction, I'm Alexander Stamers, and I'm a fourth-year PhD student in the group of Ruben van Bokstel. And we're situated in the Prinses Maxima Center for Pediatric Oncology that sits in Utrecht in the heart of the Netherlands. And we're actually the only pediatric oncology hospital in the Netherlands, which means that we're seeing roughly 600 kids newly diagnosed per year. We have over 400 researchers, and we're one of the biggest centers for pediatric oncology in Europe. Our mission, I think that's very important to always reflect back to the mission of the Institute, and our mission is to cure every child with cancer with an optimal quality of life. And I think for my project specifically, what motivates me is the second part of this sentence, so the optimal quality of life. Because pediatric Burkitt lymphoma actually has a very good success story, same as pediatric ALL that Luca previously mentioned. However, Burkitt lymphoma is an aggressive form of B-cell lymphoma and is regarded as one of the fastest growing tumors. I've asked my clinician collaborator to share some pictures, clinical images of these patients. And as you can see, they come to the hospital with these huge masses of tumor, predominantly in the abdominal region, as you see on the top, but also sometimes in the jaw region. And these children have to be treated immediately with high doses of chemotherapy rounds. Luckily, the cure rates of childhood Burkitt lymphoma have exceeded over 90%. However, this success story comes with many side effects. Because of the fast-growing tumor, a lot of these tumor cells die, and this can lead to tumor lysis syndrome, as well as hematological, gastrointestinal, cardiac, and neurological toxicities, which are mostly contributed because of the intensive chemotherapy treatment. Also, refractory relapse cases have extremely poor outcome. So what this means is that we need to enter a new phase of highly personalized and precision medicine, such as the use of small molecule drugs. But then the next question is, what do you target? And if you look at Burkitt lymphoma under the slide, under H&E staining, it looks very monotonous. It is this dense population of medium-sized lymphoid cells, which are germinal center-derived B cells, and they create this starry sky appearance. So I'm from the Netherlands. Of course, I would mention one famous Dutch painter called Van Gogh, and it's been sometimes compared to the Starry Nights painting of this painter. Since these densely packed tumor cells are interspaced with macrophages, which are engulfing these highly proliferative but apoptotic cells. However, if you look at the mutational landscape of Burkitt lymphoma, the story is much more complex than this. The Burkitt lymphoma is described by this genetic hallmark, which is the MYC-IGH translocation, which brings the IGH oncogene under the expression of an Ig immunoglobulin loci, and this leads to excess MYC protein expression. However, we know now that the genetic hallmark, this MYC-IGH translocation, is not the whole story, and that this translocation is accompanied by additional genetic alterations. And there's a plethora of different mutations and genes which have been found to be mutated in these patients, including, I'll just bring my laser, including ID3, which is an inhibitor, a negative inhibitor of TCF3, which is important for the proliferation pathway. MYC is also targeted itself, and FOXO1, for example, which are important for cell growth and protein synthesis. We have p53, which is important for survival, and so forth. So there's a wide range of genes being targeted. And you would think that since the MYC translocation itself is a genetic hallmark, all patients have the translocation, then just target MYC, right? But actually, a lot of MYC inhibitors are being tried to be used in the clinic, but so far any clinical effectiveness hasn't been achieved. So there's definitely something more going on than just the MYC translocation. And indeed, I've just shown here our own data from our own center. This includes 21 bulk whole genome sequencing tumors. And what you can see in this oncoplot, where columns are the samples and rows are the genes, that each patient carries multiple driver mutations, a mean of 4 to 6, a mean of 6 driver mutations per patient. So there's multiple cooperating mutations within each tumor, but the question is, in what order do these occur? What can we learn about the temporal order of these drivers? I want you to take a slight step back, and I want to explain to you the different kind of tumor evolution that can occur, because I think that's really important to understand before we even attempt to design more effective therapeutic targets. And there's 4 different mechanisms that have been proposed. The first one is the linear evolution model, where one precancerous cell acquires a mutation which causes the clonal sweep, so it takes over the whole tumor. And then one other driver is picked up along the way, which also then causes a clonal sweep, and so forth. So at the end, when you sample the tumor, you get this very monoclonal population. Another case is branching evolution. This is when mutations are picked up over time, but 2 or more driver mutations can cause their own expansion, and so they are sort of fighting for selection. And this means that at the time point of sampling, you can get 2 or more subclones present in your tumor. And there's 2 sort of extreme versions of branching evolution, which is mutual evolution. This is when there's no one clone or multiple or a few clones taking over, but actually each clone picks up a driver mutation, and this causes this confetti-like image here on the right. And also punctuated evolution. This is when it's also been termed the Big Bang evolution, where in the very beginning of the tumor, many different driver mutations are picked up, but only a few of them actually manage to survive, and a couple of them can be sampled at the very end. And so how do we then study intratumor heterogeneity? And I think there's 3 main ways that you can study this. One is deep sequencing. This is when you sequence very deeply in bulk your cells, but this has some caveats, because if you the idea here is that you use the mutant allele frequency to then propose or speculate what the percentage of that clone can be. But what happens if you have 2 mutations which have roughly the same allele frequency? You cannot decipher whether that belongs to the same clone or whether that's 2 separate clones. So that's the problem with ultra deep sequencing. We have multi-region sequencing. This is, I think, mostly important for maybe solid tumors, but that's the idea that you select different regions, spatial regions of the tumor, and you sequence that in sort of bulk. And you can then maybe find any differences between the clones depending on the location. But as I showed you in Burkitt lymphoma, if you look at under the H&E staining, it looks very monotonous. So where do you even start picking your cells? And you might even select regions which are on the barrier between 2 clones, if they're even spatially there. So you can never be sure you're taking pure subclones with this technique. And of course, I wouldn't be here if I wouldn't be introducing the single cell sequencing. That's the idea where you take individual cells and pick up the mutations per cell. And this avoids both the spatial bias as well as the bulk deep sequencing approach. So there's I think 2 ways where you can perform whole genome sequencing on single cells. You can take a single cell, and if that cell has an intrinsic ability to proliferate in vitro, you can then perform single cell clonal expansions and take up this and make a sort of a clonal expansion of this and sequence that. But in many cases, some of your cells that you want to sequence and analyze, they're not able to intrinsically proliferate in vitro, such as in primary material Burkitt lymphoma. And also, deriving single cell expansions can take a long time and is quite laborious. So then PTA, I think, solves this issue as well, where you take individual cells and you directly whole genome amplify the DNA of each individual cell and then analyze that further down. And I see I include the same picture that Luca mentioned in his slides before, so I won't be going into the technological part of this technique. Just to show you the experimental workflow of our single cell whole genome sequencing using PTA, we take we obtain samples from the biobank. These are in single cell suspensions already, and we use a FACS sorter to sort to enrich for both tumor cells and normal cells from the same patient. And we do this by using CD20. CD20 is a marker expressed on the cell surface of all B cells. And CD10 is a marker specifically expressed on germinal center derived B cells, which is the cell of origin of Burkitt lymphoma. So we enrich for tumor cells using CD20, CD10 positive cells, and CD10, CD10 negative cells for our normal B cells. Then we perform whole genome amplification using PTA and whole genome sequencing. So for this project so far, we have single cell sorted 332 cells from 6 patients. So I'll be going through the data in just a moment. But so we use FACS to enrich for our cells population, but we also wanted to make sure that the cells that we have sorted also match what we want to analyze. So since the genetic hallmark is this MYC IgH translocation, we first wanted to see whether the cells have this translocation or not. So here includes a screenshot from an application called IGV that allows you to read DNA reads in a very user-friendly manner. So the top row is as a single cell has been sequenced, and it derives from this CD10 positive population. And the row below, it represents the cell from the same patient, but this has a CD10 negative phenotype. And what you can see here from these reads, these orange reads represent reads which partially map to chromosome 8, which is where MYC is found, and chromosome 14, which is one of the Ig loci. And since these reads can be easily can be mapped to both, we can say that this translocation occurred here in this patient, and that this is absent in our normal B cells. So now we can group our cells into MYC IgH translocated cells and non-MYC IgH translocated cells. So our Burkitt and our normal B cells from the same patient. The first thing we want to do is to see whether the mutational load per individual cell differs between normal and malignant. So to do this, we incorporated additional healthy B cells derived from healthy donors, which had been sequenced in a Nature paper that came out 2 or 3 years ago. So we compared our study with theirs. And what you can see is that our normal B cells from Burkitt lymphoma patients, it falls nicely within the expected range of normal B cells from healthy donors. But in our MYC translocated B cells of our patients, there's a 3 times increase in mutational load between as what would be expected for that aged B cell if it was normal. So now we see that there's an elevated mutation load, but the next question is, okay, what is the clonal relationship between normal B cells and tumor cells? So that's the beauty of doing the single cell whole genome sequencing, is that you can start reconstructing the phylogenetic tree of these individual patients. And we do this by looking at what mutations are shared and which ones are unique between cells. And I just want to, before I show you the patient data, I just want to clearly explain to you clearly what I mean by this phylogenetic tree. So this is a cartoon figure, and on the x-axis in this case, we have the molecular time, which is basically the number of mutations accumulated in this cell, since we know that mutations accumulate in a linear fashion across age. And let's say we have sequenced a cell A, and this cell A has accumulated, we have found that it has 700 mutations. If we sequence another cell B in this case, we can then say that 600 mutations are shared between these two cells, but then each cell has 100 and 150 mutations privately in this case. And then if we sequence the third cell, cell C, we can make this tree even bigger and narrow down when these mutations occurred a long time. So in this case, all three cells have shared 300 mutations, and then 300 mutations are shared between only A and B and so forth. And when you overlay this information with driver analysis, you can, for example, look at, say, oh, driver event has occurred in this time point, and driver 2 occurred further on, and is only shared by a subclone of these cells. I hope you understood the principle of phylogenetic tree building. So I want to take you to a real example. So here the axes are reverted in this case, but on the y-axis we have the mutational burden, which is somatic-based substitutions, and each branch here represents the length of mutations, and the points at the end represent an individual cell. In this case, orange circles represent normal B cells from the patient, and the blue cells and the blue circles represent Burkitt lymphoma cells from the same patient. And I just wanted to point out again that these are cells derived from the same patient at the same time. So a few things pop up instantly. The fact that normal B cells diverge very early on from the tumor cells, and there's no clonal relationship between the normal B cells and what will become the tumor. We see that indeed the normal B cells in generally have a lower mutational load, and that the Burkitt lymphoma cells have a larger mutational load. In this case, we see that all these tumor cells share this trunk, which represents the pre-cancer state of this tumor. And further down the tree, you can see some intermediate clades forming, which are then followed by these clonal expansions, which occur later. And I just want to then show you the power of the single cell by comparing the tree by looking at which mutations in the tree can be picked up by bulk whole genome sequencing. Because for the same tumor, I have also performed bulk whole genome sequencing, in this case at 30x resolution. And what you can see is that if you take the mutations of the bulk of the trunk of the tree, roughly 94% of those mutations are picked up by the trunk, by the bulk whole genome sequencing. In the intermediate branches, which are these ones here, these are only picked up 40% in the bulk. So you're missing a lot of information about the intermediate branches. And of course, with the private branches, which are only specific to individual cells, the mutations which are specific to individual cells, these are of course not picked up at all by the bulk bulk sequencing, and of course also not in the wild type individual branches. So now we can, with a single cell approach, we can really look at the clonal architecture and the evolution of the tumor. Now with the same tree, I want to overlay the drivers that we have annotated. So what we can see here is that the MYC IGH location, of course, is it's part of it can be traced back in the trunk of the tree, as well as two individual MYC mutations and the SMARCA4 mutations. And what was interesting here is that this chromosome 1q gain, which if you would have looked at the chirogram of the bulk whole genome sequencing, you would say that this is shared by all tumors. That's a clonal gain, but actually we could place this copy number gain within our tree, and only a subclone of our tumor cells shared this chromosomal aberration. These tumor cells here on the left did not have this chromosome 1q gain. So we can then now say for certain that this copy number gain is a late event. And what was also interesting in this case, in this patient, was that this P10 and P2RY8 driver genes were hit twice, once in the trunk, but also downstream in only a subclade of these tumor cells, suggesting that these specific genes are under strong selection pressure and convergent evolution. In this case, the subclone, which did not have the copy number gain and the P10, P2RY8 driver genes, did have an ID3 mutation, and another known driver mutation in Burkitt lymphoma. There's another example, again, of another patient. This MYC-IgH translocation was again found in the trunk, along with p53 and SMARCA4. But what we see further down is that there's much more going on. In this case, PHF6, which is a known driver gene, is hit 2 times, one in this clade forming this population, and once again in the larger clone of this tumor. Again, showing that convergent evolution takes place, in this case on the PHF6 driver gene. Another example is this one here from another patient, which was 17 years old. One thing that really popped up immediately was the clonal how the branching formed in the normal B cells, because in my previous previous trees, I showed that all the normal B cells diverged very early on. But now in this case, we see actually the clonal expansion of the normal B cells, which is something you wouldn't expect at an age of 17 years old. Clonal expansions usually occur at a very later age. But we could actually found that all these clonal expanded normal B cells had a mutation in ACTG1 gene, which produces gamma actin protein. And this, in the study where we took the normal B cells as a reference, also noted down that this was the only gene that had a high selection pressure in normal B cells. So this could explain why we see this branching going on in the normal B cells. Again, in our tumor cells, we see the chromosome 1q gain occurring later on in the evolution of the tumor. And in this case, we see 3 different ID3 mutations taking over the tumor at the very later stage of the evolution and forming 3 separate clades. Again, highlighting that convergent evolution takes place later in the clonal evolution of Burkitt lymphoma, and in this case on ID3 driver gene. That's we also another tree where we find notch 3, for example, in this case, being the driver of the major subclone of the tumor later on. We also have the chance to analyze tumor cells from 2 different locations. So in this case, a lymph node tumor sample and a bone marrow tumor sample were taken both at diagnosis. So we were really interested to look at the spatial evolution of the tumor. And what we see in this case is that at this point in time, the 2 clones separated, and one migrated to the bone marrow and picked up later on a TFAP4 driver mutation and chromosome 4q loss, which led to the major subclone forming in the bone marrow. Whereas in the case of the lymph nodes, the PHF6 driver mutation was picked up early in the beginning, and we couldn't for now annotate what happened here, why this clonal expansion occurred, but we are looking further into our data to hopefully find an explanation of why there's a clonal expansion at this stage here. And this is a patient where we had a diagnosis and a relapse sample available. So here what was interesting in this patient was that all driver mutations that we could identify were placed in the trunk. And this led to the expansion of the diagnosis sample at this stage, and another clone migrated to the bone marrow and led to clonal expansion 6 months later. It's still very puzzling to us. We're still trying to understand what's going on. Why does this, in this case, patient not require an additional driver? It could be something about this specific cocktail of driver genes annotated here in the trunk that gives a perfect recipe for a clone to expand without needing a secondary hit. I've mentioned a couple of times that in the later parts of the tree, we could pinpoint when chromosomal aberrations occurred. So the fact that we find late timings of CMVs triggered us to see whether this timing is also present in a larger cohort. So we look back in our 21 bulk whole genome sequencing data, and I just want to show you here the frequency of copy number gains and losses across the genome. And we see that chromosome 1, and especially chromosome 1q, is a highly frequent copy number gain, as well as chromosome 7. And you can time CNVs by looking at the variant allele frequency of mutations on these gained chromosomes. So in case of a copy number neutral loss of heterozygosity, a mutation which is present in the chromosome before this gain will have a VAF of 1, a variant allele frequency of 1, theoretically. But a mutation which is acquired after the copy number, copy neutral loss of heterozygosity, will have a VAF of 0.5. The same holds true for mutations for chromosomes which gain. Again, a mutation which is acquired before the copy number gain will have an allele frequency of roughly 0.66, whereas mutations which are accumulating after the copy number gain will have a variant allele frequency of 0.33. So you can look at the ratio of the mutations before and after copy number aberrations to make a prediction, a relative timing of copy numbers. So we looked at our data, and on the x-axis here is timing, relative timing from 0 to 1, 0 being the earliest time point and late being the latest. And we see not a normal distribution. We actually see a tendency of copy number gains occurring later on in the evolution. And we looked at our recurrent copy number aberrations, as in copy number chromosome 1 gain and chromosome 7 gain. Indeed, we see consistently late copy number gains occurring in these tumors. So our single cell data could also be validated with our bulk whole genome sequencing data. The fact that we do whole genome sequencing means that we don't only look at drivers, we can also look at passenger mutations. And we can look at passenger mutations not only as individual mutations, but also in the context of other mutations. And I'm not sure whether you're familiar with mutational signatures, but it's a way of analyzing the pattern of DNA mutations, because those patterns are usually attributed to certain biological processes. And the way we look at them is using these 96 trinucleotide plots, where you look at the mutation of a base, but also keeping into consideration the bases that have occurred before and after this mutation. And by looking at the frequency of these peaks, you can then decipher what process could have led. So, for example, the C2T mutations occurring in the CPG context are known to be clock-like signature. They are due to the spontaneous deamination of 5-methylpsilocin. But tobacco smoking leads to a different pattern, SPS4 in this case, and this is mostly attributed to C2A mutations. And exposure to ultraviolet leads again to another signature, in this case a T2C mutation in the context of having a GTT context. So now we can actually look at the mutational signatures and even contribute and find the relative contribution of the signatures within our tree. So now I've shown the same tree as before, but in this case, on the branch, each branch shows the relative contribution of a certain mutational signature. And we could find 6 signatures back, SPS1 and SPS blood, which are clock-like age-related signatures, which are intrinsic. SPS7A, which is extrinsic, as is caused by ultraviolet radiation. SPS9 is known to be caused by somatic hypermutation, which is a process that occurs in the germinal center B cells. And SPS17B and SPS18 are reactive oxygen species related signatures. And I just want to pinpoint the power of this. We have looked back at the sample where we have a lymph node and a bone marrow sample at diagnosis, and in the private branches of the lymph node sample specifically, we found a high contribution of SPS7A, which is the ultraviolet signature. And this could be explained by the fact that the lymph node that was sampled was one that was taken from the neck of the patient, which is a region that could be exposed to ultraviolet radiation. But besides that, we wanted to look at the temporal dynamics of the mutational processes. Specifically, what are the processes that occur during the early development of the tumor, so the pre-cancer state? What are the processes occurring at the intermediate early Burkitt lymphoma state, and what happens late in the Burkitt lymphoma in the private branches? And what was interesting, particularly, is this SPS9 temporal dynamics. SPS9 is a somatic hypermutation signature, as I mentioned, and this seems to be active both in the pre-cancer state, but also in the early intermediate cancer state. But it drops drastically in the Burkitt lymphoma private branches in the late stage of the tumor, suggesting that this mutational process is important in driving the early evolution of the tumor, and could also be maybe the one contributing to the acquisition of the driver mutations, but this process is lost at the later stages. So the take-home messages is that in our lab we were the first to look at lymphoma to study the genome of the lymphoma at single cell resolution. And we initially found that there's an increased mutational rate in the tumor B cells compared to the normal B cells. We have uncovered the presence of intratumoral heterogeneity, and this is really in the field, I think, very groundbreaking, because it's been always thought that this MYC translocation is the event, and these cooperating events are also initiating the tumor, but once the tumor starts expanding, that is it. But we actually show that that's not the case, and that additional drivers accumulate at the intermediate stages, which cause clade-specific expansions. And the fact that we see driver genes hit multiple times within the same tumor could also suggest that there's evidence of convergent evolution. So this points towards a branched evolution model. If you remember back in one of the initial slides I presented, and perhaps even the punctured evolution, it could be that in the very beginning of the tumor, there has many different driver mutations being picked up, but only the strongest survive, and those are the ones that we pick up with our single cell sequencing. Those 2 or 3 or 4 clades that we have found. And it was also interesting to find that the MYC translocation, as well as the MYC, p53, and SMARCA4 mutations, were always found in the trunk of the tree, suggesting that these are important for the initiation part of the tumorigenesis, but not necessarily for the clonal expansions, which occur later on. And so, to really go back in the very start of my presentation, the fact that we want to find better targets. Targeting these genes and pathways which are under selection pressure, i.e. the ones which are picked up in the intermediate stage of the evolution, could be the ones that are actually the Achilles heel for this tumor. And maybe these are the ones that effective therapeutic targets can be developed. I want to finish with acknowledgements, of course, my mentors Ruben and Federica for their support, as well as Lorian, Mark, and actually another Mark, who are important who helped a lot with the wet lab and dry lab part of this project. And with that, thank you, of course, your attention, and I'm happy to answer any questions that you may have.
Thank you, Alexander. Excellent presentation, very interesting topic. And I invite the audience to place some question in the Q&A. But meantime, maybe we can address some of the questions that I have. For example, in this study, have you used also other omics approach? And why then you select really for the DNA aspect?
Yeah, so the DNA aspect was chosen because of the fact that we see in Burkitt lymphoma many mutations occurring, and it's not driven by just the MYC translocation, but additional drivers. So we wanted to see when are these picked up along the tumor evolution. And for the resolve-own part, for this project, we haven't applied it. We are planning on applying it for a different lymphoma called Hodgkin's lymphoma. And the reason why I want to use it there is because we know that Hodgkin's lymphoma has a very important microenvironment role. It uses the T cells around it to really form a shield. So we want to look at the RNA to see whether any specific receptors or ligands are expressed by certain groups of cells in the tumor. But for the Burkitt, we didn't, at least for now, go for the resolvome, mainly because the microenvironment and the receptors seem to be very important. Although saying that, we do see some clades in our trees which we cannot place a driver upstream. but they could be caused by epigenetic changes. So looking at the RNA as well could give the answer as to why, for example, this relapse clone expanded 6 months later. Why, even though it didn't pick up a clear genomic driver, it could be an overexpression of a particular gene that has led to the expansion in the relapse. So for now, not, but maybe in the future.
Okay, thanks, Alexander. We have one question from the audience. They're asking if you used Base Jumper to analyze your data or you developed your own pipeline.
Yeah, so we do use our own pipeline. It's called potato. It's not the vegetable. It is PTAPO. Yeah, and then we also have published a Star Protocols paper where we also give tips and tricks as to how to perform the pre-whole genome sequence, whole genome amplification steps, so the sorting, the QCs, as well as the post-sequencing steps. So I would recommend having a quick look at the Star Protocols paper if you want to look at more details on how we perform our pipeline.
Yep.
Cool.
Thanks. I know that paper, and I will share with the audience after that. Thanks.
Thanks.
I think, Luca, I've got one more question coming up. So, Alexander, what the selection pressure looks like during the evolution of the tumor, does it change?
Yeah, so the fact that we found drivers in the trunk and the intermediate taxes, but not in the private, we wanted to look at also the selection pressure. So what we did was we performed a DNDS ratio. So you look at the ratio of non-synonymous changes, so protein altering changes versus synonymous changes, so changes that lead to the same protein sequence. And we actually see that in the trunk and in the intermediate branches, there's a DNDS ratio above 1, which suggests a positive selection. So drivers are positively selected. But actually in the private branches, surprisingly, our DNDS ratio falls below 1. So not only is it neutral, but actually there's a selection pressure to avoid getting more drivers. Maybe the tumor itself has realized this is a perfect combination to really expand, and we don't want additional drivers to being picked up later on, which would compromise this expansion. So yeah.
Okay, that's great. I think we have time only for one more questions because there are two questions. Both of them, they're asking how many cells did you analyze, and also what was the strategy to sort the cells? Was like individual cells or like 10 cells per well, and
Yeah.
how satisfied are you with that?
Yeah, so for this project we have sorted single cell sorted 332 cells, which have gone through the initial QC. So what we do, we basically sort using the FACS sorter individual cells in particular well, so one cell per well. And the cell then already sits in the PTA buffer, which is then worked up using the PTA kit. And once you whole genome amplify, the next step that we take to QC is we perform a multiplexed PCR, which is it's a kit that randomly these are primers that randomly bind to different regions of the genome. So you can tell whether there's a good overall amplification of your genome before you send for sequencing. So from those 332 cells, 80 of them failed this QC, and we kept 250 cells, which were then shown as today in the presentation in the trees. So in our case, roughly 20% failed from our QC steps. We are quite stringent in our lab. We want the best quality cells. We think that's the best approach. But yeah, I hope that answers and then how many cells do you sequence? That's always a reviewer question. You can sequence a lot. But if we compare our trees with collaborators also from the Sanger Institute, which is our main collaborators, they also sequence roughly 60 to 80 cells per tree. It really depends on your research question. I think with that number of cells, you can really tell a lot about the subclonal structures because you will pick up the major clones. Of course, you cannot be certain that you pick up all the clones. There might be some which are not picked up, but I think the major clones will be picked up just by random chance, especially if your sorting strategy is unbiased. Of course, if your sorting strategy is biased, then you'll get more questions because maybe the markers you've used to sort the cells might enrich for a specific subclone of cells. That's why we chose to go really broad and only sort for CD20, a pan B cell marker, and CD10, which is a known germinal center B cell marker.
Yeah, that's great. That gives like a better overview on how to select the strategy for sorting and the cells that you are picking.
Yeah.
So I think that we can wrap up. Thanks again for the excellent presentation, Alex. Olivier, thanks a lot for organizing and supporting the webinar. So I want to thank you everyone to join the webinar. It will be, the recording will be placed on the Proteigene website.
Thank you. And of course, if you have any questions after this meeting, I've put my email address, or maybe contact Luca and then he can forward you to me. But thanks a lot for inviting me again. It's been nice. And yeah.
It was great.
Thank you so much.
Thanks, Alexander. Thanks, everyone.
Thank you, everyone.
Bye-bye.
Bye, everyone.
Bye.
Have a great day. Bye.